Hello, good evening, and welcome to the next presentation for UIL Biology. In this presentation, we're going to go through example questions. As always, you can email me at biomcgehee@gmail.com to contact me if you have any questions over any of the content. Okay, so I want to first start out with kind of where we ended with the previous presentation, and that is looking at the hints. And remember that there are 10 main topics. Usually the questions, there's going to be 2 from each of the topics and probably not in the same topic area. And so I tried to spread those out across the topics. And then of course as the tests progress, especially at the state level, the topics categories of where we could categorize one of those questions becomes a little bit blurred. But right here is where I really want to begin to address the example questions. So sometimes the questions will actually piggyback on content from other tests. And so one study tip here is to try to determine why the incorrect answers in the question, even if you got it correct, why those incorrect answers are wrong. And so they might be completely out there, which is sometimes the case, or the question might be related and there might be a specific situation and when it is correct, or perhaps the original question could be reworded. And so that would be some level of thinking that you would need to do in order to address those. Also, if you don't recognize some of the words that are in the incorrect answers, you should definitely look those up because they might show up on a future test. Okay, and just a reminder, bolded words in textbooks are helpful. They create a foundation. Once you have that foundation, which will get you into the invitational tests, then understanding the concepts around those bolded words will increase your knowledge in that particular content area. And then this year, again, we're going to focus on the eukaryotes as the disease, and there will still always be an in the news. So let's take a look first at what I mean by piggyback questions. So for example, in an invitational B, and I think these were questions from the 2019-2020, from the 2018-2019 session. So in invitational B, the cis face of the Golgi apparatus generally faces towards the, so here we are, invitational B. We're asking specifically about the Golgi apparatus, or we're in cell biology here. And we're asking about one side of the Golgi apparatus. So there are actually 2 sides, the trans and the cis side. So the cis face is actually always pointed towards the nucleus. Okay, now the piggyback comes from the district test. So the next test from invitational B would have been district. And this question asks, modified proteins leave the Golgi apparatus in secretory vesicles, do so on which side of the Golgi body? And so this is going to be the opposite side, which is the trans side. And so a student who is faced with this question on the invitational test would need to understand, perhaps they didn't get the question right, they didn't know what cis meant, or they didn't know which side was which. Perhaps they got it correct, but regardless, they would need to look up Golgi apparatus and look at the extra, if there were any other faces of the Golgi and what those were responsible for, to understand what was happening at the district level. Here is another example. Invitational A, some RNA molecules can catalyze chemical reactions. These are collectively called, and the answer here is ribozymes. So a catalytic RNA is a ribozyme. So a student then would take, at some point, the district test, and here is a piggyback question. The ribosome is classified as a, and here are the choices, and the actual correct answer here is ribozyme. So in this case, we've looked at ribozymes twice, but the difference is there's a word that looks very similar, ribozyme, ribosome, and so it would be easy on the invitational A to perhaps get those messed up. But if a student was looking at invitational A and were reviewing this question, they would need to know how ribosomes potentially fit into this particular question, because it showed up again 2 tests later on the district tests. So because the RNA portion of the ribosome is what catalyzes the peptide bond formation, it is actually an example of a ribozyme. Okay, so let's take a look at a couple of more sample questions. And if you recall from the previous presentation, there are 3 levels of question type. So what I'd like to do is just focus on one topic at a time, and at that point move through 3 potential levels, or level 1, level 2, level 3 of potential questions for this particular topic. So our topic that we're looking at is relationship between structure and function, and that could include basic biochemistry, cell biology, biological membranes, membrane transport, structure and function of organic macromolecules. So in our first question here, this is going to be an example of a level 1 question, something you might find on Invitational, or perhaps even this particular level 1 you might find on Invitational, or perhaps on District. Probably not a level 1 question that you would see on regional or state though. Okay, so the question is, phospholipids are found in, and so the student needs to understand what phospholipids are, but also where they are located. And so you're going to find phospholipids in membranes, and that would be the correct answer. So very basic question. If we move on to a level 2 question, we're also going to be talking about membranes here, but it requires a little bit more thought. So molecule X is polar and at a higher concentration within the interstitial fluid than within the cytosol of a cell. Movement of X into the cell will likely occur via, and there are several options here. We have active transport, simple diffusion, facilitated diffusion, secondary active transport, and a pump. So we need to, first of all, looking at the answer choices, we need to decide if this is a form of active transport or if this is a passive transport mechanism. And so we're looking at where the concentrations are. So molecule X is polar and at a higher concentration within the interstitial fluid than within the cytosol. So the interstitial fluid, even if the student doesn't know where that is located, it is referencing that that is the location outside of where the cytosol is. So this must be the external environment. And so if molecule X is higher in the external environment than within the cell, movement of that into the cell will occur via, and that's what we're trying to figure out. So we're moving from an area of high concentration to an area of low concentration. So this is actually going to be a form of passive transport. And so instantly we can remove active transport, secondary active transport, and then students may not know yet that a pump is actually an active transport mechanism. It uses energy. So active transport, secondary active transport, and pump, all of those answer choices can be eliminated. So we're down to 2, simple diffusion and facilitated diffusion. And so at this point, choosing between simple diffusion, which would be the movement of a molecule directly across the membrane without using any kind of protein transport system, a protein that's embedded within a membrane to transport, rather, or facilitated diffusion, which is going to have a protein, a transporter that's made of protein that's embedded within the membrane to assist. And so the difference between these two is what type of molecule is being moved. So it says molecule X is polar. So polar molecules have a very difficult time diffusing across membranes on their own, and that's because of the hydrophobic nature of the very interior portion of the membrane. And so polar molecules will always require an assistant, and we call that a facilitator. And so in this case, the answer is actually going to be facilitated diffusion. And so I categorize this as a level 2 in general because this could come up, I would classify this as a level 2 maybe on a district test, because this requires quite a bit more thought, quite a bit more linking of different concepts. I would not categorize this as a level 2 on an invitational. If this showed up on an invitational, which I think it probably would not, but it could, it would probably be more of a level 3 on an invitational. Okay, and then an example level 3 question. I'm referencing secondary active transport here using the sodium and potassium pump. So the question is, the transport of the secondary molecule glucose is linked to the sodium-potassium pump. If primary export of sodium could be blocked, which of the following would occur? So in this particular question, a student is going to need to know how the sodium and potassium pump work. And they're not going to be given any other reference to that. And so a student is going to need to know that potassium is going to be pumped into a cell and sodium is going to be pumped out of the cell. And that's the primary active transport, and that's the part that's going to use ATP. Glucose transport is actually linked to sodium-potassium pump. And so glucose and potassium can be symported back into the cell. So sodium can come into the cell, and in that process, it's going to bring glucose in with it. And that means that the glucose transport is linked to the concentration of sodium. And so in this case, in this hypothetical example, we're going to say if primary export of sodium could be blocked, if we can block the export from the cell in the primary active transport mechanism, what would happen to the cell? And so our choices are glucose would decrease inside the cell, more ATP would be hydrolyzed, potassium would increase in the cytosol, and sodium would easily diffuse across the membrane. So we're going to instantly kick this one out. Sodium would easily diffuse across the membrane. And that's because sodium is a polar or charged molecule, not polar necessarily, but charged molecule here, and it's not actually going to come across the membrane very easily on its own. We can address potassium would increase in the cytosol. So again, this answer choice to either confirm or reject it, to accept or reject it, the student is going to need to know more about the sodium-potassium pump. This is why we're in a level 3 situation. It's really drawing on a lot of background information. And so if we blocked the primary export of sodium, potassium would increase in the cytosol. And that's not true because we are exporting potassium, we are at the same time importing, we're exporting sodium, but at the same time importing potassium. So if sodium is blocked from exporting, we're not going to be able to import or increase the concentration of potassium in the cell. More ATP would be hydrolyzed. Again, we can kick this one out because with the sodium-potassium pump shut down, there's no additional ATP that's going to be hydrolyzed for the energy source. So that leaves this one. Glucose would decrease inside the cell, and this is the correct answer. And that's because if we blocked primary export of sodium, we'd no longer have the sodium gradient on the outside of the cell in order to use as an energy source. We're going to use that gradient, which was built using ATP, so it's a form of stored energy. We're going to use that gradient to bring in glucose. And if the gradient is non-existent, then glucose could not come into the cell. So the concentration of glucose in the cell would decrease. Okay, moving on to our next set of sample questions. We're going to look at the topic of gene expression. And in this topic, our subtopics include protein synthesis, regulation of gene expression, and the effects of different mutations. So our example here of a level 1 question, we have just simply a mutation that replaces an adenine with a cytosine in DNA would be called A. And since we're replacing a base, this is a type of point mutation, and specifically since we're replacing, it's going to be a substitution. So a very basic, probably would have seen a bold word in the textbook about substitution or point mutations and the types of consequences for point mutations. In this one, since we are not inserting a base, there wouldn't be a frame shift or deletion or an insertion. Since we're not deleting, we wouldn't be also introducing a frame shift. So as we are looking at this particular question, I can kind of break this down and say, okay, well, there's a mutation that replaces an adenine with a cytosine. We know that it can't be frame shift, deletion, or insertion. Deletion and insertion are going to be addressing what's happening at the DNA level. Frame shift is a result of a deletion or an insertion as it impacts the protein concentration, or the protein being made in translation, rather, sorry. Substitution, if we had a reversion, reversion is a much higher level of a type of mutation where cells can actually revert that point mutation either directly or indirectly back to a functioning protein. So that's a higher level type of mutation. So we're going to stick here with our basic substitution, which is exactly what's happening when we replace that adenine with a cytosine. An example question, level 2 or 3. So I've combined this into level 2 or 3 because it depends on what type of test it's on. This definitely requires quite a bit of thinking. There's a little bit of a process involved in that. So if it were on earlier test, a district test, for example, or before, would probably be a level 3. But as we get up into the region and state test, I don't know that I would keep this as a level 3. I think this is probably a level 2. I think a level 3 there could be a much harder question that could come from this. And so therefore I think for those higher tests, region and state, I would probably categorize this as a level 2. Okay, so the example here, the question is compare the 2 DNA template sequences and determine the consequences of mutation. So we have 2 sequences. We have a wild type sequence, which is the unmutated, and we have the mutation. And so what we're looking at, and it's one of the key words in this question, is template sequence. So we have a DNA molecule that serves as a template for generating the messenger RNA. And so our messenger RNA is always going to start with 5', and it's going to be complementary to our template. And so our messenger RNA for a wild type would be A, T, G, and then, I'm sorry, we're talking about RNA, so A, U, G, and then U, 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 and then U, A, U, and then C, G, and U. And that would be our messenger RNA that's coming from our wild type sequence. If we do the same thing with our mutation, we're again going to have A, U, G, U, 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 A, and then C, G, U. And we're going to end with 3'. And so we can immediately identify where the mutation occurred. So we know that AUG is going to be a start codon. Then we have UUU on both of those strands. But then we have, for our wild type, our next codon is UAU. But for our mutation, our next codon is UAA. So there was actually a base substitution in that third base of UAU that will convert that to a coding codon, so one that codes for an amino acid, to a UAA, which is actually a stop codon. Okay, so whenever we have a base substitution that generates in the final messenger RNA a stop codon, we will end up ultimately with a truncated protein. And so this is clearly not a silent mutation, otherwise there would be no impact to the protein sequence. This is actually going to be a nonsense mutation because anytime a stop codon is introduced, that is called a nonsense mutation. And I remember that because sometimes you hear people say, stop the nonsense. And so in my mind, that is how I've linked the introduction of a stop codon to this particular term, a nonsense mutation. A missense mutation would have changed a codon from one that codes for one particular amino acid to one that codes for a different amino acid. And then frame shift and deletion kind of go hand in hand. A deletion would lead to a frame shift, and a frame shift would change up the entire coding for the remainder of the protein after the point that was mutated. So this particular example, our answer is going to be B, which is nonsense. You'll note down here that a genetic code table would be provided in most cases, and that's true. In this particular case, it would be easy for students to figure out that UAA is a stop codon. It's only one of 3. And so therefore no genetic code table was provided for this example. if the codon went from UAU to something that also codes the same amino acid, I would have provided a genetic code table so that students could actually determine that it was a missense as opposed to a silent mutation. That's about the only way, unless somebody has memorized a genetic code table, to actually figure that part out. A frame shift you could figure out just by counting the number of bases in the mutation and comparing that to the number of bases in wild type. And same for deletion. If insertion had been an answer, counting would also help you with that. Okay, an example question for level 3. A mutation was introduced within the gene that codes for peptidyltransferase activity of the ribosome. Select the most immediate effect of this mutation. So key here, select the most immediate effect. So while all the answers could occur, we're wanting to know what would happen first. What would be the first issue that the cell would run into? So in our question, a mutation was introduced within the gene that codes for peptidyltransferase activity. So a student needs to know what peptidyltransferase activity is. And peptidyltransferase activity of the ribosome is the catalytic activity of the ribosome. This is the part that actually forms a peptide bond. So if a peptide bond can't be formed, then the most immediate would be peptide bond formation would cease, and that would stop. Some of the other answer choices, transfer RNAs would not be able to bind to the ribosome. That could become true, but not immediately. That would become true because there would be transfer RNAs waiting, and there would be nothing else that could bind to the ribosome until that transfer RNA exits. The ribosome would not translocate. That's also an effect. This means that the ribosome would not move down the messenger RNA molecule. But again, it's not going to be an immediate effect. Codons would be prevented from binding to anticodons. Again, that goes hand in hand with what's happening up here in A. And then if we look at answer choice E, the two subunits of the ribosome would dissociate. That would also occur, but much later. And so we'd have a stalled ribosome because peptide bond formation wouldn't occur, and that would mean the ribosome is no longer moving, and there's a specific set of events that occur whenever a stalled ribosome occurs, and one of them or could end in the two subunits of the ribosome dissociating and finishing, or not finishing rather, translation. So looking for those keywords, things like immediate effect, and then also knowing peptidyltransferase, these are going to require students to consider what is actually happening and to draw on previous knowledge. Okay, so that concludes this presentation on example questions. Again, if you want to reach out to me, my email is listed on the UIL's website, biomcgehee@gmail.com. You're welcome to ask me questions, maybe look at old tests and ask me questions about that as well. So I look forward to working with you in this UIL season. Have a good evening.